Hello students, in this video we are going to see about vitamin-like substances. So if you see the composition of this multivitamin tablet, it contains benfotiamine, which is a form of thiamine. It contains mecobalamin, form of vitamin B12, and pyridoxine. So in addition to this vitamin formulations, there is a thing which we can see here, that is alpha lipoic acid. So this thing is not a true vitamin, but it is a vitamin like substance. So we are going to see about such vitamin like substances in this video. So what is a vitamin like substance? They are basically organic compounds related to the vitamins in activity. In function, they will be related to the vitamins, but we cannot define them as vitamins because mostly these vitamin-like substances will be synthesized by the humans adequately. So they are not required in the diet. So one of the criteria for vitamin is it is a micronutrient which should be needed in the diet. But these vitamin-like substances, mostly they will be synthesized by the humans adequately. They are not required in the diet. Only in special or certain disease conditions, we will give them as supplements. So, they are classified with B complex vitamins due to the similarity in biological function and the distribution in the foods. So these vitamin-like substances, they are similar to the biological functions of vitamin B, and the sources also somewhat similar to the vitamin B complex vitamins. So they are classified with B complex vitamins. So what are the vitamin-like substances which we are going to see today? First is choline, other one is lipoic acid, the next is inositol and para-aminobenzoic acid. These four vitamin-like substances are going to be discussed in this video. First, coming to choline. So it is a trimethyl derivative of hydroxyethylamine, or we can say it as trimethylethanolamine. So here we have three methyl groups. So trimethylethanolamine. So from where we are getting this ethanolamine? So the serine undergoes decarboxylation. The amino acid serine, which is derived from glycine, undergoes decarboxylation to form ethanolamine, which will be subsequently undergoing methylation. So this methyl will be, the CH3 will be derived from the one carbon pool. So it will be methylated 3 times to form trimethylethanolamine or choline. So this choline is a significant part of one carbon metabolism. So what are the sources? Plant sources, cereals and vegetables; animal sources, liver, milk and eggs. So what are the functions of choline? First, it is a lipotropic factor. What is a lipotropic factor? So they prevent fatty liver. How they are preventing fatty liver? They prevent the accumulation of triglycerides in the liver. So this choline is required for the formation of phospholipids, which helps in the production of VLDL, very low density lipoprotein. The function of VLDL is to mobilize the triglycerides from the liver to the periphery. So triglycerides will not be accumulated in the liver. So obviously it prevents the fatty liver. So Choline is a very good lipotropic factor. Next thing is it contributes in the cell membrane structure. So we studied in the cell membrane structure that it has phosphatidyl-choline. So it contributes in the cell membrane structure. And next important function is choline in the form of acetylcholine is a very important neurotransmitter, and it also contributes in the 1-carbon metabolism and transmethylation reactions. So it is required for the production of S-adenosyl methionine, and in this property it resembles the vitamin B9, that is folate. So the next vitamin like substance is lipoic acid. So it is apparently essential for certain microorganisms, but in case of mammals no deficiency is seen till now, and it is not a true vitamin. So the structure of lipoic acid is it contains sulfur. So it has 2 sulfur. So the structure is 6,8 dithiooctanoic acid. So here we can see the 2 sulfur molecules, and it is fat as well as water soluble, and the oxidized form is known as lipoic acid, and when it is reduced it is known as dihydrolipoic acid. So what are the biochemical functions of lipoic acid? So it acts as a cofactor for oxidation reduction reactions. It is similar to thiamine in its coenzyme function. So the vitamins, most of the vitamins, they act as coenzymes in many important reactions in the body. So this vitamin like substance lipoic acid also acts as a coenzyme, and that coenzyme function is similar to thiamine. And the reduced form of lipoic acid, it provides thiol group for the reaction in the vitamin K cycle. So the vitamin K cycle has the enzyme epoxide reductase. So the reduced form of lipoic acid, it provides the thiol group and helps in the formation of vitamin K, and it acts as a very good antioxidant. So how it is similar to thiamine? So it acts as an coenzyme for pyruvate dehydrogenase enzyme complex, alpha keto glutarate dehydrogenase complex, branched chain alpha keto acid dehydrogenase. So all these enzymes have thiamine as their coenzyme, and lipoic acid also act as a coenzyme for these enzymes. And how it is a good antioxidant? It reduces the free radicals, stimulates the glutathione production, that is a very good antioxidant, and it decreases the insulin resistance and LDL levels. So the LDL is notorious for the fatty deposition in our body. Due to its antioxidant property, we can use it for treating patients. So the therapeutic uses is when we are giving it at a high dose, 100 to 600 milligram per day, it reduces the oxidative stress, that is the antioxidant property. So it reduces the free radical attack and increases the glutathione formation. So ultimately it acts as a very good antioxidant. So the next vitamin like substance is Myo-Inositol. So it is also known as vitamin B8. Its structure is a hexahydroxycyclohexane. So it structurally resembles sugar alcohol. So it is basically a carbohydrate, and it is similar to the structure of glucose. The sources are plant sources, fruits, vegetables, whole grains and yeast, animal, muscle, brain, erythrocytes and retina. So the synthesis of myo-inositol, it takes place in two steps. So the glucose 6-phosphate present in our body undergoes isomerization by the enzyme inositol 3-phosphate synthase to form myo-inositol 1-phosphate, and this undergoes dephosphorylation by inositol monophosphatase to form myo-inositol. So this synthesis occurs mostly in kidney and testicles. So what are the functions of this myo-inositol? Again, it is a lipotropic factor, prevents the fatty liver, and the cell membrane has phosphatidyl inositol. So it contributes in the structure of cell membrane, and in addition to its presence in the cell membrane, it also acts as a second messenger by being converted to inositol phosphate 3. So this picture shows how the phosphatidyl inositol present in the cell membrane act as a second messenger. So second messenger is when a hormone reaches the receptor. So here the hormone which we are seeing is insulin. So insulin, it attaches to the receptor present in the cell membrane. It activates the phosphatidyl inositol present in the cell membrane. So it will be converted to PIP3. So this form will activate the further signal transduction pathway. So which results ultimately to the usage of glucose inside the cell. So the insulin brought a message, and that message was carried further towards the interior of the cell by this phosphatidyl inositol. So we call it as a second messenger. So suppose in case of insulin resistance, this mechanism will be impaired. So this inositol will be supplemented, and it will be beneficial in conditions where the insulin resistance is more common. So like polycystic ovarian syndrome, where the insulin resistance will be more common. So supplementing inositol, myo-inositol for such patients will be beneficial. So clinical significance. In case of diabetic patients having peripheral neuropathy, there will be decreased inositol in the nerve fibers. And the phytic acid, which is a hexaphosphate derivative of inositol. So it is present in the plant-based food. So the plant food is rich in phytic acid, which is a hexaphosphate derivative of inositol. So what is the problem with this phytic acid is, we humans don't have the activity of the enzyme phytase, which is needed for the absorption of phytic acid. So there will be low phytase activity in the humans. So the phytic acid will not be absorbed in the human body. But where the problem occurs is, this phytic acid has the tendency to form insoluble salts with the minerals which are needed for us, which should be absorbed in our digestive system, like calcium, iron, magnesium. So when we take a food with increased amount of phytic acid, not only it is not absorbed in the human body, in addition to that, since it forms an insoluble salt with calcium, iron and magnesium, it also results in the poor absorption of these minerals. So the patient will have poor absorption of calcium, iron and magnesium due to the interference by this phytic acid. So which is a hexaphosphate derivative of inositol. So the next vitamin-like substance is PABA, that is para-amino-benzoic acid. So it is like B complex vitamins. It is a water soluble compound, and it occurs extensively in our nature. And some microorganisms like bacteria, they require para-amino-benzoic acid for their growth. and it is a part of the folic acid molecule. In this picture we can see the structure of folic acid molecule. It is made up of pteridine, para-aminobenzoic acid and glutamic acid. So this para-aminobenzoic acid is present within the structure of folic acid. So the para-aminobenzoic acid itself a vitamin like substance. So since it is present within a vitamin, we call it as vitamin within vitamin. So it is an organic compound with benzene ring having a side chain of amino and carboxyl groups. So the clinical significance of para-aminobenzoic acid. So we have sulfur drugs which are structurally similar to para-aminobenzoic acid, structural analog of para-aminobenzoic acid. They are nothing but para-aminobenzene sulfanilamide. So this structural similarity helps us to interfere the folate mechanism in the bacteria. So this para-aminobenzoic acid will be converted to folate. So that's why in the previous slide we saw that this bacteria need para-aminobenzoic acid for their growth and survival. So when we are targeting this conversion by a structurally similar compound, that is sulfur drugs, to interfere with this conversion, to stop this conversion by the enzyme dihydropteroate synthetase, the bacterial growth will be limited. So this sulfur drugs will become antibiotics. They inhibit the growth of the bacteria. So this is a clinical significance of the structural analog of sulfur drugs, which is structurally similar to para-aminobenzoic acid. So to summarize, we have seen 4 vitamin-like substances. The first is choline. So it has 3 methyl groups. It is a trimethyl ethanolamine. So it helps in the methylation reactions. It contributes to the 1 carbon pool, and it is present in the cell membrane as phosphatidylcholine. It is a lipotropic factor. It prevents fatty liver, and it is a very important neurotransmitter in the form of acetylcholine. So the next vitamin-like substance is lipoic acid. So it is a cofactor in the oxidation reduction reactions in the body. The coenzyme function is similar to thiamine. It is an antioxidant, reduces the oxidative stress, increases the glutathione synthesis, and it provides thiol group for vitamin K cycle. Next vitamin-like substance is inositol. So it is present in the cell membrane as phosphatidylinositol. It acts as a second messenger by being converted to IP3, and it also acts as a lipotropic factor. The derivative form phytic acid, it is excessively present in the plant-based food. It is not absorbed in our human body, whereas its presence in the food, it interferes or decreases the absorption of calcium, iron and magnesium, and it is structurally similar to glucose. Basically it is a carbohydrate. It is synthesized from glucose 6-phosphate, and it is also known as vitamin B8. And finally we have seen about para-aminobenzoic acid. So the sulfur drugs, which are structurally similar to para-aminobenzoic acid, they interfere with the conversion of para-aminobenzoic acid to folate in the bacteria. So it reduces the growth of the bacteria. It acts as an antibiotic. And this para-aminobenzoic acid present in the structure of folic acid. So it is also known as vitamin within vitamin. Thank you.